It's a big day for small things. This year on October 7, 2024, the Nobel Prize in Physiology or Medicine was awarded to two incredible scientists, Victor Ambros and Gary Ruvkun. Their groundbreaking work uncovered a whole new world of control happening inside our cells. They discovered something called microRNA, which are tiny pieces of genetic material that act like master switches for our genes. Think of it this way: our DNA is the giant cookbook for building and running our bodies. For a long time, we thought we knew the main ways the kitchen staff, our cells, decided which recipes to use. Ambros and Ruvkun showed us there was a hidden layer of managers, the microRNAs, making crucial decisions we never knew about. So, what exactly is this microRNA? Let's break it down. You've probably heard of DNA, the famous double helix that holds all our genetic blueprints. To use a recipe from this cookbook, the cell makes a copy called messenger RNA, or mRNA. This mRNA copy then travels to the cell's protein-making factories. For decades, scientists thought this was a pretty straightforward process. But Ambros and Ruvkun discovered that it's not so simple. They found these super short strands of RNA, the microRNAs, that don't build anything themselves. Instead, their job is to find specific mRNA messages, and, like a diligent editor, either block them from being read or mark them for destruction. It's a form of quality control and timing, ensuring proteins are made only when and where they're needed. This process is called post-transcriptional gene regulation, which is a fancy way of saying it controls genes after the initial copy is made. Imagine you send an email with instructions. The person receiving it is about to follow them, but just then, a tiny sticky note appears on their screen telling them to ignore the email. That sticky note is the microRNA. It doesn't change the original DNA recipe, but it stops the final product, the protein, from ever being made. This discovery added a completely new dimension to our understanding of how life operates, revealing a subtle, elegant system of gene tuning that happens in every one of our cells. Before this discovery, the scientific community largely believed that gene regulation happened primarily at the DNA level, with proteins called transcription factors acting as the main on and off switches. The idea that another type of RNA molecule could play such a powerful regulatory role was completely unexpected. It was like discovering that a symphony orchestra had a whole section of musicians you'd never seen or heard before, quietly directing the tempo and volume of the entire performance. Ambros and Ruvkun's work opened up a new chapter in genetics, proving that the cellular world was far more complex and intricately managed than anyone had previously imagined. Their persistence turned a scientific curiosity into a fundamental principle of biology. So, how did they find this revolutionary molecule? The story begins not with humans, but with a humble creature, a tiny transparent roundworm called Caenorhabditis elegans, or C. elegans for short. These little worms are a biologist's dream because they are simple, develop quickly, and scientists can track every single one of their cells from birth to adulthood. In the late 1980s and early 1990s, Ambros and Ruvkun were studying genes that controlled the timing of the worm's development, the genetic clock that tells a larva when to become an adult. They were both focused on a particular gene called LIN4, which was known to be a key player in this developmental timing. Victor Ambros's lab was trying to figure out what the LIN4 gene actually did. After a lot of hard work, in 1993 they finally isolated it. But what they found was a total surprise. Instead of a long strand of RNA that coded for a big, complex protein, they found two very short RNA molecules. They were too small to be a recipe for any protein they knew of. It was a real head-scratcher. This tiny RNA didn't seem to fit the standard rules of genetics. At the same time, Ambros had already shown that LIN4 worked by shutting down another gene, called LIN14, but he didn't know how. The discovery of this tiny RNA was a clue, but the puzzle was far from solved. Meanwhile, Gary Ruvkun's lab was investigating the same mystery from a different angle. He was studying the LIN14 gene, the one that LIN4 was supposed to be controlling. He confirmed that when LIN4 was active, the protein made by LIN14 disappeared. But here was the weird part. The mRNA for LIN14 was still there. The cell was still making copies of the recipe, but the final dish, the LIN14 protein, was never being served. This was a direct contradiction to the way everyone thought gene regulation worked. It meant something was stopping the process after the mRNA was made, but before the protein was built. The big aha moment came when the two labs compared their findings. Ruvkun and Ambrose realized that the sequence of the tiny Lin4 RNA was a perfect mirror image, or complementary, to a small section on the Lin14 mRNA. It was like finding a key that perfectly fit a lock they didn't know existed. They proposed a brand new mechanism. The tiny Lin4 microRNA was physically sticking to the Lin14 mRNA, blocking the protein-making machinery from reading the message. At first, many scientists thought this was just a weird quirk of worms. But in 2000, Ruvkun's lab found another microRNA, LET7, not just in worms, but in flies, fish, and even humans. Suddenly, this wasn't a worm thing, it was a life thing. This intricate system of microRNA control is essential for our bodies to function correctly. These tiny molecules help guide processes as fundamental as an embryo developing its organs, our immune system telling friend from foe, and our cells dividing in an orderly fashion. They are the microscopic traffic cops directing cellular activity, ensuring everything runs smoothly and on schedule. But what happens when these traffic cops are asleep on the job, or start giving the wrong signals? When microRNA regulation goes haywire, the consequences can be serious, leading to a wide range of human diseases. The delicate balance maintained by these molecules can be easily disrupted. For example, cancer is often a story of uncontrolled cell growth, and it turns out that microRNAs are deeply involved. Some microRNAs act as tumor suppressors. Their job is to put the brakes on cell division. If the gene that produces one of these microRNAs is mutated or deleted, the brakes are gone, and cells can start to multiply without limit, forming a tumor. Conversely, other microRNAs can act as oncogenes, or cancer promoters. When these are overproduced, they might shut down the very genes that are meant to protect us from cancer. Scientists have now linked specific microRNA patterns to many types of cancer, including lung, breast, and prostate cancer. It's not just cancer. The influence of microRNAs extends to nearly every corner of medicine. In heart disease, for instance, certain microRNAs can contribute to the thickening of heart muscle or the formation of scar tissue after a heart attack, impairing the heart's ability to pump blood effectively. In metabolic diseases like diabetes, specific microRNAs can interfere with the body's ability to respond to insulin, the hormone that controls blood sugar. Even our immune system relies on them. Dysfunctional microRNAs can lead to autoimmune disorders, where the body's defenses mistakenly attack its own healthy tissues, causing conditions like lupus or rheumatoid arthritis. The list goes on. Researchers have found that mutations in the genes that code for microRNAs can cause specific inherited disorders, such as certain types of congenital hearing loss or skeletal abnormalities. There's even a condition called Dicer-1 syndrome, caused by a mutation in a protein that's essential for producing mature microRNAs. This syndrome significantly increases a person's risk of developing various types of tumors. All these examples paint a clear picture. The discovery of microRNAs wasn't just an academic curiosity. It opened our eyes to a fundamental cause of human suffering and gave us a whole new set of clues to investigate in our quest to understand and fight disease. The journey from a tiny worm to a Nobel Prize is a powerful reminder that fundamental science, the kind driven by pure curiosity, is the engine of progress. The discovery of microRNA has completely reshaped our understanding of life's complexity and has thrown open the doors to incredible new possibilities in medicine. Before Ambrose and Ruvkun, our map of the human genome was missing a crucial layer. We saw the genes, but we didn't see the tiny, powerful regulators that fine-tune their expression. Now we have a much more complete picture, and with it, we have brand new targets for treating diseases that have long stumped doctors and scientists. This new knowledge is already being translated into action. Because microRNAs are so specific and powerful, they make for very attractive therapeutic targets. Imagine being able to design a drug that could restore a missing tumor-suppressing microRNA or block one that is promoting disease. This is no longer science fiction. Researchers are actively developing therapies based on microRNAs. For example, anti-mRNA drugs could be used to shut down a harmful microRNA that's overactive in a cancer cell or a diseased heart. Conversely, miRNA mimics could be introduced into cells to replace a microRNA that's been lost, helping to restore normal function. Furthermore, microRNAs are proving to be excellent biomarkers. A biomarker is a measurable sign in the body that can indicate a disease or condition. Because diseased tissues often release specific microRNAs into the bloodstream, A simple blood test could one day be used to detect cancers or heart disease at a very early stage, long before symptoms appear. This would be a game changer for diagnostics, allowing for earlier and more effective intervention. It's like having an early warning system for our health, all thanks to these tiny molecules that were first discovered by studying how a microscopic worm grows up. So, the next time you think about what makes us tick, don't just think about DNA. Remember the microRNAs, the tiny but mighty regulators working tirelessly behind the scenes. The Nobel Prize awarded to Victor Ambros and Gary Ruvkun celebrates not just their brilliant insight and perseverance, but the profound impact their discovery is having on all of our lives. It's a beautiful example of how asking a simple question about a simple organism can lead to answers that change the world, paving the way for a future where we can treat diseases with a precision we once only dreamed of. It's science, and it's awesome.